Good morning everyone, thank you for joining us today. Today we'll be discussing Sclerotinia stem rot in canola with Kira Beard from our Geraldton office. Over to you, Kira.
So we're going to be speaking about Sclerotinia stem rot, and this disease can infect canola, lupins and other broadleaf hosts, including wild radish and capeweed. But today we're going to be looking at Sclerotinia stem rot in canola. So this is what we're going to be talking about today, the two pathogens that cause Sclerotinia. We'll talk about which crops are affected by Sclerotinia, and we'll be focusing on Sclerotinia sclerotiorum. We'll look at the life cycle and how that is important for understanding how to control the disease. We'll talk about the sclerotes surviving in the soil for up to 6 to 10 years and how you can use fungicide sprays and when the right conditions occur for the disease to be a problem. So the use of fungicide is particularly relevant when infection occurs during flowering after significant ongoing rain and about 75% relative humidity. So Ravjit's found you need about 40 mils of rain before flowering for the disease to be a problem, but in the northern ag region this may not be quite that much needed for the disease to be a problem. Spraying at 30% bloom is considered economically effective if conditions are right weather-wise, and generally it's best not to spray before 10% bloom or after 60% bloom. But we'll talk about this more in the next few slides. Sclerotinia sclerotiorum and Sclerotinia minor. So in the picture here on your right, you can see this is the sclerotiorum, and it's usually, that's the main way that you can tell the difference between the two pathogens, is that the size of the sclerotes are quite different. So the sclerotiorum has large sclerotes, usually seen on the stem and inside the stem, and minor has smaller sclerotes, which are seen in white cottony growth on the outside. So these pictures show Sclerotiiorum, which are large rat dropping size, and then the minor are much smaller, and you can see in this picture. So generally minor is not considered common in broadacre areas, so we're focusing on Sclerotiiorum today. So having a look at the life cycle, this is a good guide to show you where the places are to consider the risk of getting Sclerotinia in a canola crop. So we'll start at the bottom. Here in summer, the Sclerotia can survive for many years in the soil, and they're the way that the disease is harboured over in the soil. So they can germinate under the right conditions in the autumn or winter when the weather is wet and the right kind of humidity. So usually when the canopy in the canola crop closes over, there's enough humidity underneath for these apothecia to be produced. So that's when the sclerote germinates. It forms this little white mushroom, and eventually over time it turns kind of a orangey brown colour as it ages. So these little apothecia produce millions of ascospores. So they can travel for kilometres, and you only need like one apothecia really to infect a whole paddock, because they produce each one produces so many spores. And we found in our research that they can regerminate. So they last about a month, and then another one will replace it from the same sclerote. So they're quite clever in the way that they're designed to keep spreading the disease if the moisture in the soil is continuing. So then these spores can infect the petals, and the petals are like an energy source for the pathogen, so that if the wet and humid conditions occur during flowering, if conditions are right, then the infected petal can infect the stem when it falls down into the canopy falls onto a stem or a leaf, it can infect the crop, and this leads to the lesions that we see on the stem. They can start off quite small white lesions, and then they develop into severe, long, elongated parts of the stem that are white. And then if you broke open the stem later in the season, you could find sclerotes inside, which are the way that it continues the cycle. So this outside cycle is the Sclerotiorum one, and the direct germination of Sclerotia to infect the stem base, that can happen through minor, so Sclerotinia minor. So that's not very common, and we haven't had any evidence of that, but overseas they have. So the main cycle we look at is the outside one at the moment. So as I said, the Sclerotiorum Sclerotia, they look like rat poo. So this is a picture of me holding some. So they're quite easy to spot when they're in the canola seed, because they're a bit bigger than the canola seed. But on the ground, obviously, they're hard to spot if you're looking for them underneath the crop. So they can survive in the soil for years, and only those in the top 2 to 3 centimetres of the soil would germinate. And the conditions that are needed for them to germinate are ongoing moisture of like 7 to 10 days and cool temperatures of like 10 to 20 degrees Celsius. So this is a picture of some healthy looking apothecia that have germinated from one single sclerote, and then over time this is the same ones that I took a photo of a month later, looking very shriveled and dead looking, so they're on the way out. So the interesting thing is that we're doing some work looking at sclerote germination, and we've got the trial at Esperance, Albany, South Perth, Northam and Geraldton. So DAFWA are looking at what conditions favour the Sclerotia to germinate. So this is the second year we've done it, and the same result has been seen both years so far. In the Albany, they germinate first, they germinated in June this year, and early June, and then a month later they were germinating in Northam. And the other sites haven't seen them yet, but we've had reports through PestFax of them being seen at Gibson and Albany, so in June and July. So that's a sign that the conditions are right in the soil for these to be germinating. And obviously depends on the soil type as well, because heavier soils will stay wetter for longer, so they're more likely to come up first in heavier soils and wet areas. So just to give you an idea of how we score disease, so if you're looking at doing a trial, you might like to consider this, or just to help you see what the stages are. This is the first stage you might see. We don't always see this, but sometimes you can see the leaves get affected first. If a petal that's infected falls on the leaf, it causes this watermark symptom that you can see. Then the next severity score 2, is when the leaf that has that watermark symptom, it starts to rot and it will break, it will bend in half like that, and you'll see fluffy growth growing on it. Then the infection spreads to the stem, or in some cases it just starts directly at the stem, and the small lesion that you might see earlier in the season becomes a long, elongated area of the stem that's white and often fluffy if it's moist and humid. Then the stem starts to rot and sclerotes are produced inside it. So if you broke the stem open, you might see those inside. And then the whole crop where the infections are will start to stand out because it's really quite bleached. The plants become very bleached looking and start to lodge. So it's quite easy later in the season to spot the infected plants a mile away. So how does the disease spread? Well, as we said, it's soil-borne. When the sclerotes are continued over in the soil, can stay there for up to 6 years, depending on if they germinate or not. And the stubble contains the sclerotes inside it. So rainfall is a big determinant of when the sclerotes germinate and whether there's flowering if there's rain during flowering, and that determines what kind of years are a big year for Sclerotinia. So Ravdik Kangur has done some research into what triggers spore release, and she's found that you need at least 40 mils of rain in the 3-week period before and also after bloom, and at least 75% relative humidity. So we're still looking into this, but that's a good guide, that you need a fair bit of rain before and during early flowering for this disease to really get going. So in terms of looking at the risk factors, obviously if you grow a lot of canola in rotations, it's going to be a higher risk, and if you have an infection one year, then the disease is likely to hang around for a few years in the same paddock. And even if it's lupins, because it also gets infected by the same pathogen, it is a higher risk. So it's important to rotate non-host crops and avoid sowing downwind or close to last year's infected crop, because as I said, the apothecia can produce millions of spores. So we'd like to use clean seed, use foliar fungicide, and it's a good thing that there's lots registered, so there are lots of possibilities. And as I said, the main factor to consider is crop flowering time, and that coinciding with the rainfall. That's the main factor where the risk is quite high if there's a lot of rain during flowering. So just to make sure we're all on the same page, the flowering stage is assessed by looking at the main stem. So TAFWA work has found that on average 30% bloom in most years is the best time to apply fungicide. Now obviously it's good to be flexible and monitor future rainfall patterns, so if it hasn't rained much before 30% bloom, then you might be worthwhile putting it off until it does rain. So some years we find around 50% bloom is the better time to put it on, because it hasn't rained much until that point and the disease isn't likely to get going. So it's just a matter of being wise and considering what's happening with the weather, and also looking at the timing of the crop in question, because the time of sowing and the variety type and whatever the flowering window is will vary from paddock to paddock. So 30% bloom is 20 or more flowers open on the main stem, and that's the usual time in most years, which is a good time, but we're continuing work on looking at that. And we're also this year looking at different products to see if there is any difference in control between the different products that are currently registered. So application before 10% flowering is generally not recommended, because the disease won't have had time to get going, because it requires the petals to drop. So I think that's about it. So for more information, you can go to our website at Managing Sclerotinia Stem Rot. Canola is a great source of information, and we'd love you to report any finds to PestFacts to assist with everyone knowing when it's out and about. And we also report some of our trial results in there as they come in, but later in this year we'll have more results available for you. Thank you.